μια ερώτηση.
Εντάξει, προχωράμε. Καλημέρα και από μένα. Να καλέσουμε τώρα την επόμενη ομιλήτρια, την κυρία Αθηνά Βέρβερη, παιδίατρο και κλινική γενετίστρια, με πολύ πλούσια εμπειρία σε γενομική ανάλυση και σε κλινική γενετική, τόσο από την Αγγλία, από το NHS, όσο και από τη Μονάδα Σπανίων Νοσημάτων και Γενετικής του Νοσοκομείου του Παπαγεωργίου, η οποία θα μας μιλήσει για τα γενετικά αποτελέσματα στην πράξη και ειδικότερα για το τι κάνουμε σε περιπτώσεις αρνητικών αποτελεσμάτων, τι κάνουμε σε περιπτώσεις, πώς διαχειριζόμαστε περιπτώσεις με ευρήματα αβέβαιης κλινικής σημασίας, καθώς και ευρημάτων παθολογικών, πιθανώς παθολογικών, τα οποία όμως δεν σχετίζονται με την ένδειξη του φαινοτύπου. Κυρία Βέρβερη.
Κυρία Βέρβερη, προς τιμή του δεν είναι αδύνατη, δεν είναι εδώ. Παρακολουθήσα, αν συνομιλία στα αγγλικά.
Ναι, το οποίο τώρα το αποφασίσαμε, so you have to bear with me because I'll try to speak in English, but I prepare to speak in Greek. So I'll try. I haven't prepared anything in English. Okay. Thank you very much. My job today is to talk about the difficult results and how we can best navigate these. So we start with negative result. What's next? And in order to discuss about negative result, we have to start with what test our patient has done. So it could be whole exome sequencing, where you look at all the genes that are in our exome, or clinical exome sequencing, where you look at the genes that are relevant, that are disease-causing at the moment, or a targeted panel of genes where you have selected the genes that are relevant to your phenotype. And one question, because we talk about panels in people with myocardiopathy, is whether we have done a virtual panel or a set panel. So when we talk about a set panel, this means that we look at particular genes and we only record these genes. So this means that we look at these genes, but we cannot expand our analysis if we need to in the future. Whereas when we do a virtual panel, this means that we have a whole exome or a whole genome as a backbone. We look at the specific genes, we filter these genes that we want to look at in 2025, but then we can expand to a larger panel or even to whole exome if we need in the future. And one question is whether we go for a set panel or for a virtual panel with a backbone of whole exome. And when we can do it in terms of financial, it's usually better to go for a virtual panel in order to be able to do your reanalysis in the future. So when it comes to reanalysis, why we need reanalysis? This comes from Panel Lab Genomics England. So this is where lots of labs get we get our panels from. So this is the panel for hypertrophic cardiomyopathy, and this is like a crowdsourcing tool where lots of geneticists can log in, and you can suggest a gene that you think is relevant to this panel, to hypertrophic cardiomyopathy, and then you upload the evidence, and then there is a team which will look at this, and they will validate the gene and include it in the panel, which means that you have the genes which are green, which are the validated genes. They are relevant to your phenotype, so you include them in the panel. And then you have the amber genes and the red genes. The amber ones are where you have insufficient evidence, and the red ones are the ones with very insufficient evidence. And then you get these curations. So this guy says, I suggest this gene, but I'm not sure if this should be included in the panel. This means that when they will look at the panel in a couple of years, they may decide to include this, so the amber gene will become green gene. And this means that if we have done a panel in this patient, we can reanalyze the panel and we can include this gene because we have the backbone of the whole exome sequencing, so we can expand our analysis. And the next question comes to what our backbone is. Is it whole exome or whole genome sequencing? And the difference between these two, when we have whole exome, this means that we look at the exons only, so the important parts of our genome, which give you the RNA and the protein. And then you have the introns, which are the little links between the exons, which do not give you protein, but they do include a few mutations. So some of our patients, and I'll come back to this later, will have intronic variants, and we will miss them with the whole exome sequencing. So if you think that our genome is an apple, with the exome you get a slice, with the whole genome you get the whole thing. And what it offers us in terms of diagnostic yield the whole exome sequencing cannot pick up copy number variants, or at least it's not yet validated to pick up copy number variants. And this means that we have the mutations, the intragenic mutations, like adenine has changed to guanine, but we also have deletions and duplications in our genome. So a whole exome may have been removed, and this is pathogenic. And we will not be able to pick this up in a safe way with the whole exome sequencing. We need the whole genome sequencing here. So how common is it to get a deletion in HOCUM? We think, that's very old data, that it could be like 1% of our patients, but it's still important, and it could be much higher now that we are starting to do whole genome sequencing when we are picking up more deletions. And this is an example of a patient that we missed in our lab with whole exome sequencing. So this guy had a filament C deletion. We saw failed confidence, so we saw something, but we were not entirely sure, so we kind of dismissed it. But we asked him to do whole genome sequencing, and luckily for him, he lives in the UK, so he did the whole genome sequencing there for free, and they picked up exactly the same thing, but that was obviously very safe result. So he has a filament C deletion, which was dismissed because it came up as failed confidence in our whole exome sequencing. And coming to intronic variants you can sometimes see in the whole exome, but you can very well see in the whole genome. So we know that 2% of our negative HOCUM patients will have MYBPC3 intronic variants, and 9% of our negative HOCUM patients in general will have a positive whole genome result, mainly because of intronic variants. So again, it makes sense to sometimes go for whole genome sequencing when we are ready to do this and it's financially possible. Triplet expansion. Triplet expansions means that you don't have a mutation inside the gene has opened and it has become longer, and for this reason you have the condition. So you can't see this by whole exome sequencing. And it's not super relevant to my cardiomyopathy, but we do have a few conditions like myotonic dystrophy 1 and 2 and Friedreich ataxia, which will be picked up by whole genome sequencing or by dedicated test for myotonic dystrophy, but they won't be picked up by whole exome. And with myotonic dystrophy, we usually get the diagnosis, in my experience, because you have a baby who is very severely affected. We had one this summer in Thessaloniki, and then the mother came, and she has a cardiomyopathy, and she has a hand grip. So mom has, the grandfather has the condition, but they didn't know about it until they got this baby who is intubated for 5 months in the NICU. So you get lots of different symptoms. Between them, you get cardiomyopathy as well. The same with Friedreich ataxia. It will probably come as a referral from neurology because they have ataxia, but then you get the cardiomyopathy as well. Mitochondrial DNA, again, you won't be able to see very well with whole exome sequencing, but you will see very well with whole genome sequencing, or very well, quite well. So mitochondrial is the DNA that women pass on to the kids, so it only comes from the egg. And this is a family which had a delayed diagnosis because we didn't suspect mitochondrial DNA. Sorry, I speak quickly and I speak in English. So we had this little girl who was 8 years old and she had ataxia. We did whole exome sequencing, we didn't pick up anything, and then the family said that the mother was 35 years old with deafness and postpartum cardiomyopathy. Thoma, you probably know her. Because then they're under your care now at Ahepa Hospital. So this lady was very severely affected in terms of her heart. And then she said that she has lots of people in the family with heart problems and neurologic disease, and they have died of this. So we then suspected mitochondrial, and this family has MERV syndrome. And again, it was missed by the whole exome sequencing. And the other things that that are different between whole exome and whole genome are methylation pattern and mosaicism, which are not relevant to cardiomyopathy, so I won't go into this. But what I wanted to say is that whole exome and whole genome are not the end of the journey, because now we are getting even more clever tests, which is the long-reads NGS, which hasn't come into diagnostic praxis yet, and it's not very relevant to cardiomyopathy, so I won't go into a lot of detail. But with long reads, what you do is that you don't break the DNA into small fragments and then put them back in the puzzle to read the DNA. You break the DNA into large bits. So it's much safer to do the puzzle when you have big bits compared to small bits. So the normal whole exome sequencing or whole genome that we do is the small bits, and the long reads is the large bits. So you get safer results. when it comes to particular genes in cardiomyopathy like titin or MYAL2, because titin is a huge gene, because MYAL2 has a pseudogene, so you're not sure if the sequence that you're seeing is the real gene or the pseudogene, but with long reads you're much safer. But we don't have very good results yet in cardiomyopathy, so people are still considering it as a research, and we are not really doing diagnostic for cardiomyopathy. It has started as diagnostic in a few neurodevelopmental conditions. So when we have a negative result, what do we do next? Sorry, this is in Greek. We need to think if we have covered all the relevant genes. We need to think if there could be a deletion or duplication. And if we think there is a deletion or duplication, we can go for a dedicated test like MLPA, or you can go for whole genome sequencing, like the patient with filament C, where they picked up the deletion by whole genome sequencing. We need to think whether, based on the family history and the phenotype, it could be a triplet expansion, like myotonic dystrophy or Friedreich ataxia. We need to think whether it could be mitochondrial, again, based on the family tree and the phenotype of the patient. We need to think whether it could be non-genetic, a phenocopy or a polygenic condition. And we already know that 34%, for example, of the heredity in HOCM patients has to do with polygenic models. So this brings us to what you mentioned. So we have the polygenic, where you have lots of variants with a small impact, and then you have the oligenic, where you have few variants with a larger impact, and then you have the monogenic, where you have one variant, and it has a straight relationship with the phenotype because it has a large impact. But again, I'll come back to this because we do know that lots of the penetrance is very different between families. So there must be something going on even in the monogenic conditions. So this is something that I think diagnostic labs have to gradually get into. And we say that we can do a bigger test. So if we have a negative result in our patient, what we do obviously is reanalysis every 2 years, which is quite difficult for any diagnostic lab, I think. And then you can think about doing a bigger test, but at least in the NHS or in countries where the genetic test is covered by the NHS, we have to think that in order to do the bigger test, you want an incremental diagnostic yield of something like 10%. And then you can ask your patient to have the further test, obviously when this is covered by the NHS. And coming to variants of unknown significance, so navigating the uncertainty. Matt told us about this, so I won't go into this, what a variant of unknown significance is. But this is a bold prediction for human genomics by 2030. So the National Human Genome Research Institute of the US say that we will be able to do diffusing, so we won't have any more variants of unknown significance in 2030. So they are thinking that we can tackle this problem of variants of uncertain significance by various ways. One way is doing functional studies. And sadly, it's difficult to do functional studies in Thessaloniki. I have tried many times, and I know Yannis has tried many times. It's not easy to find someone to do a functional test for your patient. I just want to show this patient with Friedreich ataxia and cardiomyopathy. And luckily, some people from the hematology clinic at the NHS hospital in Thessaloniki managed to do an mRNA level measurement last week for a patient with Friedreich ataxia and normal genetic test. And we did pick up, she's the one with the, in the pink, we did pick up that she has a homozygous mutation. So we know she's affected, but we haven't found the mutation. Family segregation studies, so this is a big discussion. We've discussed THOMAP before, because you need a lot of people for family segregation studies, like at least 6 informative meiosis. And this is very difficult when you have incomplete penetrance and age-dependent penetrance. So normally, this is a family tree where you have 6 people. So you have this lady down who is the proband and she is affected and positive genotype. Her brother is negative and normal phenotype, and then her mother, and then the grandfather, and the uncle. So here you do have the family segregation that you want. It looks like 6 people are too much, but this is a real family tree that I had in London. And to be honest, I thought 6 is too much, and I thought that we don't need to call all these people and bring them to clinic and ask for samples. But it has happened to me that when I was sure that this was going to be segregation, we got this guy who had the phenotype and was negative for the genotype. It could be something irrelevant, but it also could be that your variant of unknown significance is, at the end, it becomes benign. Then we have to have good databases. So we need databases of all populations. We need databases of Greek people, of African people, which we are missing at the moment. We have lots of Caucasian people. So databases of healthy people and databases of affected people. And I think the important thing here is the biomarkers. So what we hopefully will do at some point, researchers, not clinicians, I think, will try to combine biomarkers, so imaging, circulating, multi-omic, all of them under artificial intelligence in order to get a result for your patient, for your phenotype, and for your VUS, variant of unknown significance. So I know that ophthalmologists have, from UCL, they have created a very nice platform where you can put the genetic variant and then the result of the OCT or the MRI or whatever they've done, and the electrophysiology, so something relevant to your electrocardiogram, and then this combines all the information and it will come up with a diagnosis and will say this variant of unknown significance is relevant or it's not relevant. So this is deep learning. I don't know if there's anything similar for cardiomyopathy. I did find a study which is a European initiative and kind of looks like this, and there is a paper. And I'll try to send them an email because it looks interesting, or if anyone knows about it, or if it is relevant, because this is about twin studies, I think, but it does have deep learning and variants of unknown significance. But I think the most important thing to do this diffusing is the MDT. So we need to speak to each other, and I know this is difficult. It's difficult for me. I know I don't pick up my phone during the day, but what we have to do is sit down on the same table or on a Zoom with the laboratory geneticist and the clinical geneticist, if you have a clinical geneticist, we don't have in Greece, and then the cardiologist, and try to discuss and see if our variant is relevant and what our plan is. So for example, for the patient with the filament C, it's important that the lab geneticist picked up something, and then for this reason we said, okay, is this relevant? The cardiologist said, yes, this is very relevant, and then we asked the patient to do whole genome sequencing. So this is how it starts, and you get a plan. Are we going to do whole genome? Are we going to do MLPA? Or are we going to stop with the patient and go to the family and start doing segregation studies? And coming to, I don't know, my time, am I okay with time? Okay, I'm finishing. So with pathogenic variants, it's just a couple of slides. We have pathogenic variants which are irrelevant, and this brings us to the problem with the prevalence. So this comes from the biobank study. So the biobank is a big database where they put hundreds of thousands of samples, and you have the medical records as well in the UK. So these are healthy people. So we know that there is a prevalence of 1 in 578, 1 in 251, 1 in 149 for ARVC, DCM, and HOCUM, and these are likely pathogenic or pathogenic variants. So these people should be affected. But the penetrance among these people is 1.2 to 3%. So we have a very low penetrance compared to our affected people. So when you have a proband with HOCUM and you have a variant, the penetrance in his family or her family is going to be 50%. If you have an unaffected person in a random database and you get a pathogenic variant, then the penetrance is going to be 3%. So this brings us to the huge difference in penetrance, which has to do with all the other variants and all the other factors, genetic or environmental. So the management of pathogenic or likely pathogenic variants which are irrelevant to the phenotype and were picked up by mistake or in research in a study like the UK Biobank depends on personal and family history and disease expression. And these patients should not be, these people should not be managed in the same way that you will manage the relatives of a proband who has the phenotype. And I think that's it. Thank you.
Thank you, Athena, for your excellent talk. Any questions?
Ήθελα συγχαρητήρια. Εγώ θα κάνω έτσι σαν γιατρός, σαν κλινικός γιατρός, μια απλή ερώτηση. Έχω μια ασθενή, η οποία έχει αριθμιογόνο μυοκλαστοπλάθεια, με διάφορες εκδηλώσεις, να μην τα βλέπω. Κάνουμε γονιδιακό έλεγχο και βγαίνει ένα βους, ας πούμε, στο γονίδιο που κωδικοποιεί τη δεσμοπλακή. Έρχεται ο πατέρας, ο οποίος έχει τον ίδιο φαινότυπο και έχει το ίδιο βους. Έρχεται και η κόρη της ασθενούς, της πρόβαλ, η οποία έχει φαινότυπο πιο ήπιο και έχει το ίδιο βους. Χρειάζομαι κάποια απόδειξη για να κάνω upgrade
το βους που βρήκα.
Θέλει και άλλες μειώσεις. Αυτό το συζητούσαμε με το Θωμά. Θέλει πολλά περιστατικά. Αυτό είναι το δύσκολο. So you need a lot of meiosis in order to reclassify the variant, because that's why I showed you that the family that I showed is a Lois-Dietz family. So I was confident that this is a likely pathogenic variant, and we wanted it for prenatal, and they wouldn't let me. The lab colleagues didn't let me because they said, no, we need to have 6 or 7. And then we got the last guy from Italy that was in London, and he was negative, and he had a huge aorta. So the whole scenario was wrong. So you do need lots of people, because it's a priori it's 50-50. So 50-50 could happen.
Η πιθανότητα πάντως τυχαίας μετάλλαξης είναι 1/2 ή στον αριθμό των μειώσεων. Δηλαδή 1/2 στην πρώτη είναι 50%. 1/2 στην έκτη είναι...
Αυτό, να πεις 1/2 επί 1/2 επί 1/2 για 6 με 8 μειώσεις, και μετά λες ότι έφτασα σε μια πιθανότητα που πλέον μπορώ να το κάνω likely pathogenic. Γιατί αυτό που λέτε εσείς, οι 2 στην οικογένεια, είναι 1/2 είναι...
1/2 στην τρίτη.
Ναι.
1/2 στην τρίτη, 1/2 στα 8.
Παίζει, αυτό να είναι λάθος. Δηλαδή, όταν βλέπεις και χιλιάδες ασθενείς, σε κάποιον θα...
Γι' αυτό χρειάζονται πολλές τέτοιες αναφορές. Αλλά πάντως είναι λογικό αυτό που λέτε, αλλά δεν καλύπτει τα κριτήρια που έχουν οριστεί και δεν καλύπτει και την ουσία του πράγματος.
Και επίσης εδώ, επειδή έχεις incomplete penetrance, αναγκαστικά θα δεχτείς και κάποιον ο οποίος έχει το γονότυπο και δεν έχει το φαινότυπο.
Ακριβώς,
θα τους βάλεις και αυτούς μέσα, ειδικά στη μυοκαρδιοπάθεια. Δηλαδή στα νευρολογικά είναι πιο εύκολο το segregation από ότι σε αυτά.
Ο κύριος Βασιλικός.
Good morning. Thank you very much. One stupid question. You've mentioned that everybody has their own data, and this data should be mixed at some stage. First of all, there is no, according to my knowledge, there is no unified way of merging data, I think, still. For example, if your center asks to merge your data with you, there may be some differences in the way that the data are stored. Second and biggest problem, the legal problem, I'm not aware, because we merge our data with the New York University, for example, and nobody, I asked my lawyers, so what should we do? And we just signed an MOU, but I'm sure it's illegal. So what is your vision? I know this is the future, okay? For example, Professor McKenna needs our data, we need his data. At some stage, there should be a universal data bank, right? So what is the legal procedure for this, or what is the thinking? Yeah, I mean, I know there are many projects, but what are you going to do?
One thing is that you can submit your variant. So when we do a lot of work and we realize that our variant is pathogenic because we've done family segregation and we have tested 10 different people and we managed to reclassify this variant, then we can submit just the variant, not the whole data, the whole VCF that Mata was talking about, the whole genome, the raw data. You can just put in clean var that this variant, we did the work, we did functional or segregation, it is likely pathogenic, so the next person can use the same thing without doing the whole procedure. And second, I have the same problem because we just got in Erdera the Rare Diseases Network, so we are now uploading our undiagnosed cases, and someone will reanalyze, because we are not capable of reanalyzing, to be honest, every 2 years. So this solves our problem because all our undiagnosed patients will be reanalyzed by a European network. They have lots of consenting, lots of work done on the ethical and the legal level, so hopefully it will be okay. But we did have a long talk with our scientific board in the hospital before getting into this project, and we haven't started uploading yet because the consent is huge. So it's not easy to get consent from the parents, but we are going to try, because it's our chance to have reanalysis by someone who has lots of data compared to me, who is smarter than me, and who has the time, because I am not going to do it, or my lab, to be honest. So we'll see how it goes.
Thank you very much. We'll discuss later. I think it was a very interesting topic. Και τώρα θα προλογίσω και τον